Yeah, hello everyone. My name is Ellen Kang. First of all, I'd like to thank the CCS Virtual Seminar Committee for this great opportunity. And I'm pleased to share and showcase my group's research today. A brief background of me is so I'm currently an assistant professor in the Department of Physics and NanoScience Technology Center, and I'm affiliated faculty at MSc and graduate faculty in biomedical sciences at Burnett School of Medicine. I'm running my lab named Molecular Biophysics and Nanobiology Lab. Just short bio of myself is that I received a bachelor and master's degree in physics from Seoul National University in South Korea, and I received a PhD in physics, biophysics at Brown University, and did postdoctoral training at Yale University before I joined UCF in fall 2015. So my area of expertise is in specifically like a protein biophysics, and primary research topics are molecular biomechanics of cytoskeletal protein assembly and protein-based mechanobiology at multiscale. In terms of teaching, that I've taught and developed several courses at UCF. And so at graduate level, I've taught principles and techniques in nanobiology and introduction to nanoscience and nanotechnology. And at undergraduate level, I've taught college physics too for non-major students, and also chemical and life science nanotechnology. This is a course that I co-developed with my colleagues at NSTC, and also have taught nanobiotechnology. So just summary of key accomplishments at UCF. I've secured, to my credit, 1.3 million funding. And I recently received the NSF Career Award, Biomechanics Mechanobiology program. And also I'm a co-investigator of an NIH R01 grant. Also received a NASA FSRP award, and yeah, other honor. As you can see. Okay, so I'd like to introduce actually three kind of like main projects currently going on in the lab. So the first one, before I jump on that, the actual motivation of an overall goal of my project is studying this very cool protein system which is called actin. So actin is it serves as a major building block of many eukaryotic cells, and actin polymerizes into filaments, double-stranded and helical filaments in the presence of ions and ATP. So this assembly of actin and also mechanical properties of actin drive many important cellular processes, including cell movement, force generation, and muscle contraction. But current knowledge gap in the field is that most of in vitro studies and biochemistry and biophysics studies on actin, as well as actin binding proteins, have been done only in dilute buffer conditions, so which does not reflect what's really happening inside the cells. So in our in vitro system, what we are really interested in is to understand fundamental mechanisms that regulate the mechanical, structural, and physical chemical properties of this actin cytoskeleton in complex intracellular environment. And specifically, we are interested in studying their assembly kinetics and mechanic structure. How does it really change, like when you have other intracellular factors, and also actin binding protein interaction. And lastly, mechanosensing, which is a key response that how this cytoskeleton responds to mechanical and physical cues inside and outside the cells. So the first project, this is the one that I'm currently funded through NSF Career Award. So as you can see that these are typical representative videos in real time that these fluorescently labeled actin filaments can be imaged using total internal fluorescence reflection fluorescence microscopy. And through this, that we can analyze their bending mechanics. So as you can see that the control is showing the the movement in dilute buffer condition, whereas when you have like a macromolecule crowding that is introduced in the system, that it will really generate this confined movement. So you can see that the actual filament bending is suppressed. So our main objective in this project is that we want to determine how intracellular crowding modulates the bending mechanics and conformations of filaments. So from mechanics change and also like the conformation change, like a major change that we have determined is that in the presence of this type of macromolecular crowding, that the helical twist of filament changes drastically. So when you have like this over twisted filament, that can also change their interaction with other binding partners and binding proteins. And secondly, we aim to evaluate the effects of this macromolecular crowding and osmolytes that can also like react to osmotic stress inside the cell, how they really affect filament assembly dynamics and kinetics. And so in this video, what you're seeing is that the real time, also again, like in the visualization of the assembly of these filaments. And we also like model this filament system in the presence of like various types of molecules and see like how the preformed filament and the subunit, like the actin monomer, actually interacts, because through looking at their diffusion behavior, that we can also learn like how these kinetics are modulated. So achievements so far, like that we have demonstrated these roles, like very important roles of this crowding in actin mechanics and structure regulation, and also demonstrated how filament assembly dynamics and kinetics inside the cells would be actually differ from dilute condition in vitro, as a lot of research have been shown. So this study actually gave us an idea of, because when you have like these changes in mechanics and structure, that will affect the interaction with actin binding proteins. So the major proteins that we are interested in is like a bundling proteins, and which is also called like a cross-linking protein. And this type of protein really plays crucial role in like a fourth generation, especially at the leading edge of the cell, because they can generate this type of like a tightly like a bundled structure. So by having and also testing different types of bundling and cross-linking proteins, and depending on their geometry, that we aim to investigate interaction between filaments and these regulatory proteins in crowded intracellular environments. And we also aim to determine how mechanics and secondary structures of actin cytoskeleton are regulated at the nanoscale. So this was done, like this study, the second project, is also currently actually ongoing project, is in collaboration with Dr. Laurent Titard in Department of Physics and NSTC, and also Dr. Michael Molinari at University of Bordeaux in France. So I'd like to mention that collaboration with Dr. Molinari's lab was actually made possible through this prestigious Chateaubriand Fellowship. My PhD student, Nicolas Castaneda, was recently awarded that fellowship. So that allowed him to actually carry out and perform this experiment in Dr. Molinari's lab, where they house this high resolution AFM instrument there, and also allows like us to investigate the actual mechanical properties, including Young's modulus of actin bundles in the presence of different various molecules. So this is well, due to COVID, that because the original plan was like actually Nicholas will stay there like for like a 6 months, or over, but due to COVID he had to return, so he actually was there like a half of the time, but still like was able to obtain like quite interesting data, which we are currently now working on to write a manuscript with Dr. Molinari. So with these projects, that we also demonstrated how intracellular crowding tunes and the organization and structure of these actin bundles by cross-linked by regulatory proteins. Okay. So the third project I like mention, because at the first, the initial slide that as I mentioned, that I'm a co-investigator of this NIH R01 grant. So this project is in collaboration with Dr. Travis Jewett, who is the PI of this grant in biomedical sciences, Burnett School of Medicine. And so in collaboration with Dr. Jewett, what we are studying is this bacteria, which is Chlamydia trachomatis. That is the bacteria that can actually cause this infection, and it's actually transmitted infection, chlamydia. So we are studying this protein called TARP effector. And because TARP effector has a role that it can induce actin bundles. But the actual mechanism of how this bundling occurs and also biomechanics of these bundles generated by TARP effector are not really well established. And so we aim to determine these biomechanical properties at the molecular level, of this TARP-induced bundles through our biophysical analysis. And also we are currently also working on determining the mechanosensing mechanisms of these TARP-mediated bundles. And based on our recent study, that we found that these TARP-induced bundles exhibit very unique mechanical properties that were very distinct from other well-known bundling proteins, which is the actual TARP-induced bundles would make a very flexible bundles compared to other types of bundles. And that was very interesting, because then what the question here is that how would this flexible bundle or really make that invasion efficiently? So that's our really actively ongoing work with Dr. Jewett. And really the impact of this work is that going to be a better understanding of how bacteria invade cells at the molecular level, can lead to novel strategies in the long run, to prevent infections. So with this brief outlook of my near-term and long-term goals. So near-term goals is that these are currently also ongoing work, that we aim to investigate how actin cytoskeleton dynamics, mechanics, and structure are regulated by combined intracellular factors. So not only just by one specific factor, but as we know that inside the cells, there are multiple factors together. So we'd like to really complete this phase diagram, when you have this, for example, crowding ion and just actin binding proteins together, all are they really working at synergistically, or are they having some type of opposing effects? And secondly, we'd like to determine how actin cytoskeleton responds to mechanical forces at the molecular level. And this really focuses on that mechanosensing mechanism. That is, I would say, not only just near-term goal, but that is also my long-term goal. So the long-term goals are I have this 3 points. That the first one is that as I showed you as one example, that I'm working with Dr. Jewett for this disease mechanism, right? So a lot of things that I think the current knowledge gap is that how we can really identify the molecular basis of this human disease, and including such as cardiovascular disease or neurodegeneration that are closely linked to cytoskeleton mechanics and mechanobiology. So our current hypothesis is that when you have disruptions and dysfunctions in this type of cytoskeleton, especially actin protein, then if you have that will actually lead to changes in mechanics and structure that can affect, and also that can lead to this human diseases. And secondly, which we are also have that as a long-term goal, is nanobiology. And specifically, we are interested in understanding interface between cytoskeleton and nanomaterials. And these are based off some of my ongoing collaborations with collaborators, Dr. Tanya Roy, and Dr. Swami Rajaraman in MSc. And also I'm currently collaborating with Dr. Kimi Sugaya at Biomedical Sciences and Dr. Santra on a project that are related to the neurodegeneration model. And lastly, Dr. Minjun Kim at SMU is also my collaborator for the nanobiology side. And lastly, I'd like to have like this study we are currently doing on actin, we can expand to other cytoskeletal systems such as bacterial and plant cytoskeleton, which a lot of like mechanism on their side is very well, not well understood. So those are the things that all the tools that we developed so far can be directly applied to these different systems, and that will really help us understand better of how these different types of cytoskeletal systems are regulated. So with this, I want to thank my wonderful lab members, current and former lab members, and all the collaborators and funding support. And thank you for your attention. I'd happy to take any questions.